Hi everyone, my name is Laura Michelle Castro Chavez. My topic now is hemoglobinopathies in the fetal position. Definition. Congenital diseases that result from mutation or genes in our need globin genes in the alter structure or speed of synthesis of a given globin chain. The destruction of the kinesis domain of an enzyme called hem-regulate inhibitor augments fetal hemoglobin in erythroblasts for patients with sickle cell disease. Application in medicine. The 14 needs apply to the pregnant patient with hemoglobinopathies, allows to know what the need is affect and to define those dependencies of a patient. We have a video.
Sickle cell disease is a group of inherited blood disorders in which the body produces abnormally shaped red blood cells that look like crescent moons or sickles. Sickle cells have a shorter than normal lifespan. Their premature destruction results in shortage of red cells, known as anemia. Signs of anemia include shortness of breath, fatigue, and delayed growth in children. Unlike normal red cells, which are pliant, sickle cells are rigid and also sticky. They may clump together and stick to the walls of blood vessels, causing obstruction in small vessels and subsequent reduced oxygen supply to various organs. This happens repeatedly and manifests as periodic episodes of pain called crises, which can last hours to days and may result in organ damage, especially in the eyes, lungs, kidneys, bones, and brain. The spleen has to handle large numbers of dead red cells and becomes enlarged and fibrous. Its immune function declines, making the body more vulnerable to infections. In an attempt to compensate for blood cell loss, the bone marrow tries to produce more cells and grows larger, causing bones to weaken. Other signs include jaundice, a result of rapid destruction of heme. Hemoglobin is the major component of red blood cells and is responsible for oxygen transport. The adult hemoglobin, or hemoglobin A, is composed of 4 protein chains, 2 alpha and 2 beta. The beta subunit is encoded by the HBB gene. Several mutations in HBB gene are responsible for the disease. Each individual has 2 copies of HBB gene. The disease develops when both copies are mutated, producing no normal beta globin. The 2 copies may be mutated differently, producing 2 different forms of abnormal beta subunits in the same person. Various combinations of these mutations produce different forms of sickle cell disease, but the most common and also most severe, called sickle cell anemia, is caused by 2 copies of the same mutation, producing the mutated hemoglobin S. Each copy comes from a parent. The 2 parents each carry 1 copy of the mutated gene, but they typically do not show any symptoms. This pattern of inheritance is called autosomal recessive. Hemoglobin S has the tendency to form polymers under low oxygen conditions. This process is called sickling or gelation for the gel-like consistency of the resulting polymer. As the polymer filaments grow, they eventually involve the cell membrane and distort the cell into the characteristic crescent shape. Apart from oxygen tension, the presence of other hemoglobins also seems to affect the sickling process. Normal adult hemoglobin inhibits sickling, and this explains why heterozygous parents who produce both mutated hemoglobin S and normal hemoglobin A do not usually develop the disease. Fetal hemoglobin F, which has 2 gamma chains in place of 2 beta chains, also suppresses sickling. Infants born with the condition seem to benefit from high levels of fetal hemoglobin in the first few months of life. They do not develop symptoms until the age of 6 months or so, when fetal hemoglobin levels drop. Bone marrow transplantation is currently the only known cure for sickle cell disease. It involves replacing the diseased stem cells in the bone marrow with healthy cells from an eligible donor, usually a relative. The procedure, however, is complex, and finding a suitable donor can be difficult. In most cases, treatments aim to avoid crises, relieve symptoms, and prevent complications. These include prophylactic antibiotics, vaccinations to prevent infections, pain medication to relieve pain, drugs that promote formation of fetal hemoglobin to suppress sickling, periodic blood transfusions to reduce anemia and prevent crises, and early detection and treatment for complications when these occur.
Now, pros and cons. Pros. There is the possibility of having an effect child for this in the performance of blood tests to identify couples at risk before pregnancy. Cons. Hemoglobinopathies are hereditary diseases of the blood that alter the transport of oxygen and can cause various disorders, including the death of the woman or the baby in strong cases. We have another video.
Let us discuss about hemoglobinopathies. We will start our discussion from the various types of hemoglobin in our body. We should keep in mind before discussing the hemoglobinopathies. We will discuss the main objectives of hemoglobinopathies. Our main objectives are types of hemoglobin, globin chains in fetal life and adult life, and hemoglobinopathies like thalassemia and sickle cell anemia. We should keep in mind most common types of hemoglobin before discussing the hemoglobinopathies. Usually in human body there are three main types of hemoglobin. First one is hemoglobin A, second one is hemoglobin A2, third one is hemoglobin F. In all of these types of hemoglobin there is only difference with the globin chains. In hemoglobin A there are two alpha chains and two beta chains are present. In hemoglobin A2, two alpha chains and two sigma chains are present. In hemoglobin F, two alpha chains and two gamma chains are present. Please keep in mind percentage of hemoglobin chains varies from fetal life to adult life. After pregnancy or conception in fetal life, the major hemoglobin is hemoglobin F. F stands for fetal hemoglobin. It is almost 90%. In fetal life, approximately 10% hemoglobin A is present up to 6 months, and some traces of hemoglobin A2 are present. Please keep in mind that composition of these hemoglobin remains same up to 6 months. After 6 months to the rest of adult life, composition of these hemoglobin changes. Chief hemoglobin after the age of 6 months is hemoglobin A, which is approximately 97%. Hemoglobin A stands for adult life. As in hemoglobin F, F stands for fetal life. Round about 2.5% hemoglobin A2 is present, and some traces of hemoglobin F is present, about 0.5%. Let us discuss about the hemoglobinopathies. Hemoglobinopathies mean destruction of RBCs due to problem with the hemoglobin. There are two main types of hemoglobinopathies. Thalassemia, which is the quantitative defect in beta chain, and sickle cell anemia, which is qualitative defect in beta chain.
Estudio aplicado en Ecuador. This pathology by the Ministry of Public Health must be permanent in addition because it constitutes a public health program. Must have in the diagnosis, prevention, and education program for the community. Conclusion finally. The frequencies of the variants found in the study confirm that hemoglobinopathies represent a public health problem and the importance of systematic screening programs for hemoglobinopathies that includes not only early treatment but also a genetic education and counseling program for family groups. And that's it. Thank you for watching.